Hello and welcome to GeneLab Chats. Today we'll be interviewing Dr. Barker from the University of Wisconsin-Madison. Dr. Barker and colleagues conducted a meta-analysis of Arabidopsis datasets available in the Open Science Data Repository in order to allow a more robust comparison between studies that share similarities in experimental design. In this interview, Dr. Barker describes the findings in this publication and how the GeneLab data systems and the analysis working groups contributed to his reanalysis of the open science data sets. Thanks for joining us, Dr. Barker. Could you please let our audience know the most important findings of your study?
Thanks for the opportunity to share some of my research. So the matrix meta-analysis combined data from spaceflight, radiation assays, and simulated microgravity studies that all use transcriptomics to assess the response of Arabidopsis thaliana plants to stressors related to spaceflight. We filtered this data by using the p-value in order to get a broad set of differentially expressed genes. We then created a connectivity network visualization system using pairwise comparisons that can be made between all the metadata saved in the GeneLab system. This identified similarities based on experimental design. The data was then analyzed collectively. We then used an unguided weighted gene correlation network analysis clustering system that identified clusters or groups of genes that responded to these specific stressors. Finally, we found that although there is benefit to using microarray data to increase confidence in some genetic responses, we also found that it essentially masked one of the flight response clusters. And that was revealed only when the RNAseq studies were assessed independently.
Thanks for sharing those insights with us. And my second question, how did the AWGs help assist you in this work so you could achieve your goals?
Well, first and foremost, all the transcriptional data used in this study was pre-processed by the GeneLab data processing team, ensuring that they were all processed through the same bioinformatic pipelines. The GeneLab plant analysis working group then provided detailed assessment and discussion of the various factors that were investigated during these experiments. This allowed us to group certain factors to find the main experimental features that were causing the changes in gene expression. This allowed us to link studies with commonalities in their experimental design. This network analysis allowed us to further visualize and explore the links between these factors and the resulting genetic responses observed in these studies. There are many questions that could be answered from the matrix of data that we accumulated. Through discussion with the team, we agreed to focus on the spaceflight response factor and identified a group of 5 studies that use really similar methodologies. We then found we were able to identify a consistent cluster of genes that responded across multiple spaceflight studies. To find out more, please check out our paper.
Barker, thanks for sharing your enthusiasm with us today and with our viewers, and maybe you've inspired some new people to look at plant data sets. And to our audience out there, you can learn more about this work, how to join the analysis working groups, and access all the data sets analyzed in this work with the links provided in the description section below. Thank you and have a wonderful day.